Hello students, so today we are going to discuss about B cell biology. So as we know that B cells are very important cells of our body, and the role of these B cell is to produce various antibodies against antigens. So the humoral mediated immunity mainly depends on this kind of B cells, right? So the story starts from bone marrow. Bone marrow is a very important part where you will find out the stem cells, and these stem cells will produce B cell precursors in the bone marrow. So these precursor cells are very important cells. So from these precursor cells, mature B cells are mainly produced in bone marrow. The mature and proliferate from the bone marrow and enters the bloodstream, right? So these B cells, without the exposure to any antigen, it is known as naive B cells, right? And these cells, when came into the contact with any specific antigen, here I'm using the word specific antigen, you'll find out the specific antibody. Basically, specific antigen, no term like, means there is no term like specific antigen. But why I'm putting this specific word? Because one word called specificity, you have to remember that word. So antibody produced by B cell is always specific to that particular specific antigen, right? So the story of B cell starts from stem cells. This stem cell was then converted into the B cell precursor, from B cell precursor to mature B cell, and this mature B cell when came into the contact with the antigen, particularly here we'll use the term called epitope, right? So epitope specific antibodies will be produced by this B cell. So the whole mechanism is very important to understand, right? So as we discuss in T cell biology, we have a TCR, here we have a BCR. What does it mean? B stands for B cell, R stands for receptor. So we need a certain kind of receptor here for this activation of B cell. So now we'll discuss about the BCR as well as we'll discuss about the mechanism. So how this works, let's see. So when suppose this is your B cell, and on this B cell we have a number of BCR receptors. We'll see the structure, the detailed structure of BCR later. So suppose all these surface receptors are very important. When this receptor came into the contact with specific antigen, this B cell is now converted into two types of B cells. Simply, one is known as the memory B cells, another one is known as plasma B cells. The role of memory B cell is to remember the antigenic structure, so that will helpful when the future exposure will be there. Now, if you focus on this plasma cell, so this plasma cell is able to produce various antibodies. So this antibody production is very important for the humoral mediated immunity. So for that reason, we need the activation of this B cell. So B cell activation is very important, and this B cell activation always starts when this antigen came into the contact with this naive B cells. Okay, so this binding is the first step for the B cell activation, and if we see the whole mechanism, so there are two types of B cell activation. One is called the T cell dependent, and another one is known as T cell independent. Okay, so it is a kind of a cell. So as we know that each and every cell is having the membrane. So in the membrane, you will find one monomer molecule of antibody along with different type of protein. So these all proteins are the transmembrane protein because they are embedded here in the plasma, right? Cytoplasma to membrane here. Okay, so it reaches up to cytoplasma and embedded into this membrane part. So this is known as the immunoglobulin. As we know that these are the Ig, most probably, and in the most of the case, it is IgM, monomeric IgM or a IgD. This unit, okay, the embedded antibody units in the B cell plasma membrane. So this is Ig alpha, this is Ig beta, and this part, particularly highlighted part, is IgM or IgD. All together is known as B cell receptor. And numbers of B cell receptors are present as, so see 1, 2, 3, 4, 5, like this, no? Numbers of B cell receptors are already there on the B cells. And the location, the position of this antibody is like this, no? So this antigen binding site is away from the cell. It's open at outside. So any of the antigen will able to bind here. Okay. So when the antigen binds with this particular site, that is the first step for this B cell activation. I hope this much is clear. So alpha and beta here will also help here for the B cell activation. Now what happened after this binding? This is the first part in the B cell activation, no? That's binding. So this binding is not enough for the antibodies secretion or production of the secretions. We need extra support here, and that support is of T cell, and that is called Th2. We already discussed about this Th2 cells, T helper cells, no? So we need T helper cells, we need T cells for the differentiation of your naive activated B cells to plasma cells. If we want this cell, then we need Th2. And that's why B cell always depends on T. So that kind of mechanism is called the T cell dependent. So 99% 1, and this is the 1% rare case. And this independent mechanism is not that much effective, because in that mechanism we don't need any kind of T cell, right? So now let's see. Now we are very much clear about the B cell receptor. So any of the nerve cell is having a number of surface transmembrane BCRs. No, BCR is a complex receptor having monomeric form of IgM or D along with the transmembrane alpha and beta, Ig alpha and alpha. Right. So this discussion leads to one conclusive part, that there are two main roles of this B cell in our body. Right. Let's write it down here. First role is to production of antibodies in the body by help of T helper cells, in some case, rare cases without the help of T cells, right? But second role is it acts as an antigen presenting cell also, because I told you here, so particularly in this antigen binding pocket, this antibody binds with antigen, and this binding later on it's a it activates the one pathway called endobiosis, no? So just like a macrophage, the fragment of antigen now engulf, kind of engulf, this goes inside, Endobiosis, it goes inside, and this B cell acts as a antigen presenting cell. So B cell not just only produce antibodies against that specific antigen. Along with that, this B cell will also acts as an antigen presenting cells. So B cell and T cell work very synchronizing, So T cells help B cell, B cell also helps T cells, and both will provide us this cell mediated and the humoral mediated immunity. So I hope this much is clear. Now we'll focus on this T cell dependent pathway, right? So T cell dependent means what? So here I'll divide this sheet into the two parts. So let's divide this into two parts first and then discuss this topic, right? So here one part is a B cell, another part is a T cell, because we need help of T cell for this activation of B cell. So we have a B cell here, we have a T cell. I'm mentioning here T helper cell. Okay. So as we know that, suppose this is our macrophage. And on the surface of macrophage, we know that we have a MHC class 2 molecule with the fragment of. Now when this particular macrophage came into the contact with T helper cell, no? We know the mechanism in T helper cell activation. The first signal means binding of TH cells to the antigen presenting cells, and we have a receptor. This is a TCR. This receptor is called TCR, and this receptor is MHC class 2. And here in the center pocket, we have an antigen, right? So this is the first activation. So first one here